Genes are arranged in the cell's nucleus in coiled and folded strands, called chromosomes. Genes and chromosomes also come in pairs. Cells reproduce by dividing. When a cell divides, the chromosomes split into two identical copies, so that each of the now two cells is exactly the same as the original. Each cell has two identical sets of chromosomes. Since there are two sets, it's called a diploid cell. There are two types of cell division: mitosis, which produces an exact copy of the parent cell, and meiosis, which produces the cells used in sexual reproduction. These cells have all the chromosomes in a normal cell, but only one of each instead of two. The time in a cell's life when mitosis is not occurring is called interphase. During interphase, cells take in nutrients, do their specialized jobs, and grow. When a cell is going to divide, it begins by duplicating its DNA in preparation for mitosis. The chromosomes uncoil so that the cell can read the genetic information they contain. The process of mitosis is divided into four phases: prophase, metaphase, anaphase, and telophase. During prophase, the first stage of mitosis, the chromosomes condense tightly into matching paired strands, and the nucleus begins to disappear. The matching paired chromosomes are joined together by a special region called the centromere. The strands of DNA on either side of the centromere are identical to one another and are called chromatids. In the material around the nucleus, called cytoplasm, small fibers called microtubules begin to form at each end of the cell. During metaphase, the second stage of cell division, the microtubules at either end of the cell attach to the chromatids and have a kind of tug-of-war that ends up with the chromatids positioned in the middle of the cell. During anaphase, the third phase of cell division, the centromere divides and the chromatids are pulled to opposite sides of the cell by the microtubules. The individual chromatids are now chromosomes. During the final phase, telophase, the new chromosomes move into their relaxed state and nuclear material forms around them, making two new nuclei. The cytoplasm splits into two equal parts, completing the division of one cell into two. Meiosis is the process of a diploid cell dividing in a way that produces a sex cell that has only one chromosome, instead of the two that are in a normal cell. Meiosis occurs only in the specialized cells of the reproductive organs. The cells produced are eggs in the female and sperm in the male. Since they have only one chromosome when they join, the new cell has two. Eggs and sperm are called haploid cells. They have a single chromosome of each type. Regular body cells have two of each chromosome, one from each parent. A regular human cell has 46 chromosomes. Human sperm and eggs have 23. There are two stages to meiosis. In stage 1, the diploid cell divides in a special way, producing two haploid cells. In stage 2, those two haploid cells divide, making a total of 4 haploid cells. Each of these stages has 4 phases. Meiosis starts with the diploid cell. The chromosomes first duplicate themselves. During the first phase of meiosis, prophase 1, the chromosomes the individual inherited from their father, called the paternal chromosomes, attach on the nuclear envelope right next to the equivalent chromosomes they inherited from their mother, called the maternal chromosomes. After they are lined up, special enzymes snip and cut different genes from both the paternal and maternal chromosomes and swap them between the two chromosomes. After this occurs, the nucleus begins to disappear, and microtubules begin to reach towards the chromosomes from either end of the cell. During metaphase 1, the microtubules move the condensed chromosomes to the center of the cell. The matching maternal and paternal chromosomes that swap some of their genes remain paired together. During anaphase 1, the matching paternal and maternal chromosomes are pulled apart to opposite ends of the cell. During telophase 1, the microtubules disappear and the cell cytoplasm divides. There is usually little or no pause between telophase 1 and the beginning of the meiosis 2 stage. During prophase 2, the microtubules reform and attach to the chromosomes at the centromere. During metaphase 2, the chromosomes are pulled to the center. During anaphase 2, the centromeres holding the sister chromosome strands together split and the strands separate. The microtubules pull each of the sister chromosome strands to opposite ends of the cell. In the final phase, telophase 2, the cytoplasm divides, the chromosomes relax, and nuclei form around them. The result is 4 sex cells, with half the number of chromosomes of the organism's regular cells. Also, as a result of the switching of different genes between the parent chromosome strands, each of the sex cells produced has a different genetic combination than the parent cell. This is why children who have the same parents don't all look the same. In male mammals, the sex cells undergo further changes as they develop a head where the DNA is concentrated, and a tail called a flagella that propels them through the female reproduction tract. When these sex cells are fully developed, they are called sperm. Human males produce millions of sperm continuously. On the other hand, human females usually produce only one egg a month. If the sperm and egg cell unite, the DNA contained in the two cells will produce an individual with some traits like its father and some like its mother.